Okay, we're going to go ahead and get started now. Looks like just about everybody's in here. And if anybody comes in, I'll add them in as we go along. This is AP1 Lab. It's supposed to sort of parallel the lecture component in here. It does to some extent, sometimes. You'll see in AP2 it tracks a lot better in AP2. Anyway, everything for lab is found in eLearn. We have a series of weekly assignments that we'll go through in class. This week we're going to take a look at the basic parts of the microscope and look at various human tissue types and take a look at the skin. Because I talked a lot about the skin and what it does for us. I thought I'd go over some of the parts of it. And the way the class is organized, there are 4 units. The first unit is all about the skin and tissues and stuff like that. And then there's a lab exam that is based mostly on identification. knowing the parts of the microscope, knowing the various cells, being able to identify the various slides. This is the same type of test you would have if you were on the ground, only the slide. The pictures are on the computer. The exams are all done online. Yeah, so, which is, what we've been doing for a while. We call these a practical exam because you have to be able to identify the various parts of cells or the cell types, being able to say, this is an epithelial cell, this is a connective tissue cell, this is a bone cell, what type of, is it a bone cell, is this a blood cell, is this a nerve cell, what are the little purple cells are they called, things like that. As well as be able to answer some questions on that too. And everything is contained in in eLearn. I also have a very detailed study guide for this first lab test. So the first lab test is all about slides and identification and stuff like that, identifying tissues. The remaining lab tests are strictly identification. The second lab test is all about bones. There are 206 bones in the human body, and you have to be able to identify them. The good news is that what's on the left side of the body is the same as what's on the right-hand side. So if you know an arm and a leg, you've got a lot of the bones learned already. And the test for that is strictly identification. You'll have 100 questions. Or maybe, I don't know, probably 100 questions, and just name the parts. The third test is muscles. There's only 60 muscles that were on our test. That's good. The 60 muscles involve, being able to identify them, and on some of them being able to name what their origin is, where they insert, and what their action is. Don't worry about that now, but that's the third test. And again, there's no study guide for that one because it's just a list. That's all it is. And the last test is all about the nervous system. If we were on the ground, we would actually be dissecting brains and eyeballs, and which is a shame that we don't get to do that. But the test goes over the parts that we would see in the dissection and being able to identify the various structures. Again, it's pretty straightforward. It's not like you either know it or you don't. So, so the format for the test, they will all be multiple choice exams. So, and that's before you get thinking, well, that's good. The multiple choice questions usually have about 20 or 20. I think I have 26 potential answers for each one. So it's a little bit more challenging. So but if you know it, it's easy. It's very easy to breeze through it. The other news about the exams is that it'll be like the lecture exam. When it opens up, you'll have like a 10-day window to take it at your convenience. But it will have to be done in a set time. For 100 questions, I think you have an hour and a half. If it were 50 questions, it would be an hour. So, or maybe 2 hours for 100 questions. I haven't decided yet. Most people tend to blast through it fairly quickly. The schedule for the lab is posted in eLearn. And while I realize that since we don't take a day out for the lab, even though I have it listed in the schedule, this is the schedule that we're going to follow. Now we will simply move on so we can get done. Because without taking the day out for the lab exam, we can continue moving forward. And what I like to do is start while you're prepping for your lab exam, like taking the test on the slides and tissues and stuff like that. I'll start talking about bones. And you're not gonna, I don't expect you to start working on bones while you're working on your first test, but it'll give me a chance to at least introduce it in class so that you'll have more time to get ready for it. The bone test takes time to prepare for, more than just studying the night before. And so I try to give you as much time as I can to get you ready for that test and for the muscle test too. Because these are these exams, the bone test and the muscle test, are going to have some real relevance for you in later on in whatever healthcare program you go into. No one's going to particularly care that you can identify transitional epithelial cells as different from cheek cells. But being able to identify all the bones and all the muscles that we cover, that's important, and that'll have a relevance. That'll come back and help you again and again. I just ran into a nursing student Tuesday. I was on campus Tuesday. I ran into a nursing student there that said that thanked me again for the emphasis that I had from her A&P classes with me, because it really helped her. It's really helping her in the nursing program. So it's nice to hear stuff like that. So I'm, yeah, some of this stuff is not going to be is not exciting, but some of it has a direct application. Anyway, enough of that. So the 4 exams, each one worth 100 points towards your final grade. It's only so you have 400 possible points. 360 points out of 400 is an A. That's 90%. If you're at 89.5%, I'll round you up. So make it easy for me, get above a 90, so I don't have to worry about trying to do the math. There is no final exam in here. So we just have the 4 exams. Once you're done with the exam, you're done. I don't give them back. I don't reopen them, because you're going to go on to new material. And that's just how we do it. There is, so it's very straightforward. My goal is since we don't have to take the days out for the exams, my goal is to have us done before Thanksgiving for the labs, to have everything covered, which will give you more time in lecture as we approach the end of the semester to prepare for your last exams and to prepare for your final exam, because we won't have classes. Once we're done with lab, we don't have classes. So I'm trying to get you done with enough time to prepare for the finals. And you'll find in AP2 we're done even quicker, because the labs are more focused and usually shorter in length too. So, okay, let's see. Syllabus. The syllabus is online. The syllabus explains everything that we're doing. It gives all the phone numbers and contact points for me and for my office location and for my dean. If you have any questions, the syllabus lists all the areas we're going to study. I've just gone through them already, but they're listed there. And the fact that we only have 4 exams. The good news also is in AP2, we only have 3 exams, makes it even easier. So, and we have, like I said, there's a schedule that we're going to follow. So next week, we'll do the second half of Unit 1, which will be diffusion and osmosis, mitosis and cell division in the parts of the cell. So it's all spelled out in the lab schedule. And we would follow that schedule even if we were on the ground. While I know in lecture, it's we're a little squishy on dates, because we don't know exactly when we're going to get done. Like the Monday AP1 class is the Wednesday AP1 class has finished chapter 1, but the Tuesday, Thursday classes are like 5 slides for finishing. So they're slightly ahead already. So not that they're going to start their test any sooner. Anyway, the point is that I want to how am I going to say this correctly? Well, I'll come back to that later. Anyway, the point. Okay, so we're going to actually, are there any questions? Where I'm like dying to jump into the topic area, but are there any questions on anything? It's always challenging to start the last lab of the week is in the beginning of the semester. You've heard all this stuff about syllabi and everything in the in lecture, so with no questions and no comments, I'm going to go ahead and jump into this. And the first thing we're going to look at is the microscope and the compound microscope. Now, many of you may have been exposed to microscopes before, and that's great. And I wish we could be on the ground for this, because it's hard to say. And then you turn this knob that's over here that you can't really touch right now. It's challenging to try and explain the parts of a microscope by just looking at them, but that's what we're going to do. Now, we have a compound light microscope. We call it compound because it has a series of lenses that are stacked on top of each other. It's a light microscope because it uses a light source. There are other more advanced microscopes like an electron microscope. UT has one. Phase contrast, nice high-end microscopes. Ours are student scopes. We do have a set of high-end scopes that if you take microbiology, which is still on the ground, as far as I know. There are some very nice microscopes that are used there. But for our purposes, we would use regular student scopes, and they do okay. The compound light microscope stacks the eyepiece and various what we call objective lenses on top of a body tube, and that allows you to magnify at different levels, different magnifications of specimens that you're looking at. We use light sources. We used to use 400 years ago, we used candles and mirrors as a light source. The light would reflect in the mirror from the candle, and then it would be directed up in microscope body tube. And that approach stayed that way for a long time. 100 years ago, we were using electric lights that would shine a beam onto the mirror. 50 years ago, many microscopes still came with mirrors on them and with a very specialized light source that shine a focused beam on the mirror. And then lo and behold, around 1960, microscopes started showing up with built-in lights. And that was a game changer, as they say. And then about 30 years ago, 40 years ago, the microscope started showing up with mechanical stages. The stage would actually move. You wouldn't have to move the slide by hand. And then somebody got the bright idea that why don't you install the body tube at an angle instead of being straight up and down? Because for 400 years, if you were using a microscope, you'd have to look through the microscope bent straight over it, looking straight down. Somebody put a hinge device on the microscope base about 100 years ago, so you could actually tilt your microscope. That was amazing. But now we have mechanical stages, we have multiple lenses, we have body tubes that come out at an angle. And the original microscopes, if you wanted to change the magnification, you have to go to a different microscope to use that, because you couldn't just switch them around. Okay. A little bit of history. A lot of improvements have happened on microscopes in the past 40 years. So the objective lenses are what we use. The objective lens are lenses of different magnifying power, and they collect an image. They magnify the image on the microscope slide. We have an ocular, an eyepiece, that takes that magnified image and magnifies it again. So we can go from 40 magnifications all the way up to 1,000 magnifications. We have various objective lenses that can go on the microscope that rotate around, and we have an ocular. The ocular is set at 10 magnifications or 10 power or 10x. The objectives go from 4x up to 100x. So if you take the 4x and multiply it by the 10x, that gives you 40 magnifications. The next one is 10, so that gives you 100 magnifications. The next one is 40, that gives you 400 magnifications. And the last objective is 100, which lets you magnify to 1,000 powers or 1,000 magnifications. We won't use that in AP, but you will definitely be using the 1,000 power lens in microbiology, because that's what you use to look at bacteria and funguses. Can't see viruses. Viruses are too small. You can see bacteria and funguses. So we have an eyepiece called the ocular, and we have the objectives. Underneath the mechanical stage, we have a device known as the iris diaphragm. The iris diaphragm works just like the pupil in your eye. The pigmented portion around your eye is called the iris, and this iris diaphragm opens and shuts to allow a certain amount to regulate the amount of light coming through from the light source. Because a lot of times we find that less is more. When we're looking at certain slides of tissues, sometimes less light is good because the light comes in, it's extraordinarily bright, it just blasts through the tissue, and tissue is so transparent, even with the stain, that everything gets washed out. So we turn the intensity, we narrow the field of light coming through a little bit, and we can see better that way. It sounds counterintuitive, but sometimes less light enables you to see better. Now, within the microscope itself, let me go back. This will go back to the objective lenses. The objective lenses, we have the shortest of the lenses, called the scanning lens, and it has a 4x stamped in red ink on the body of the objective lens, and has a red ring around it. And that tells you that that's 4 power. So the scanning lens gives you 4 magnifications, and combine that with the ocular, which is 10 magnifications. So scanning power is 40 times or 40 magnifications. This is what we always start with, because it gives you the biggest field of view. And so you can go look around at everything till you hone in on something. And then you can switch to the second lens, the low power lens, which has a 10x on it. And the 10x is usually stamped in yellow. It's a big 10 on it. And so 10x and the 10x ocular gives you 100 magnifications. That's low power. And in a lot of cells, a lot of tissues, that's all you need. 100 magnifications is more than adequate for us to see things and see things very clearly. And if that's still not what you need, you can turn the eyepiece one more notch, and you'll bring up the high power objective lens, which has a 40 stamped on it in yellow, or in blue, I'm sorry, in blue. 40 times the ocular power gives you 400 magnifications. However, you'll also notice that your field of view has shrunk considerably, and the image that your field of view has gotten dimmer because there's less light coming in. So when you go to a higher magnification, you may want to consider increasing the intensity of the light coming through. So just because you're looking at a smaller field. So we confine ourselves to areas between the 4x objective and the 40x objective, between 40 power and 400 power. The oil immersion lens lets you go up to 1,000 magnifications. It has a white band on it that says 100. You have to put a drop of oil on the microscope slide so that you can eliminate distortion from the air around you. You put a drop of this immersion oil on the slide, and then you lower your oil immersion lens till it just touches a drop of oil. And now we have a very consistent medium for the light rays to travel, because the air movement at 1,000 magnifications would actually distort the light rays. So we use the oil immersion lens all the time in microbiology. It allows you to see bacteria types, which, and that's great, but we don't need it in this class. We don't look at anything that requires 1,000 magnifications. Once you get into micro, you'll be looking at various types of bacteria, various pathogenic and non-pathogenic bacteria. And then the structure that holds the objectives is called the nose piece. I already said that about the iris diaphragm. So this is our microscope. This is actually, we've been using this same picture of this microscope for 18 years. So it's time we get a new one. But this still has all the parts. So the eyepiece is called the ocular. It's always set at 10x magnification, so 10 times. There's usually a little piece of wire embedded in there to use as a pointer, and that pointer is very useful to when you're looking for a specimen. The body tube is angled, comes out at an angle for us, which is great because when you spend hours and hours using the microscope, and you don't have to set bent over, it does make life a lot easier for you. But to make the body tube work, we have a prism in here. The prism bends the light rays. The prism bends the light rays that are coming up from the light. Here's the light source here. The light rays come up through the slide on the stage and into the objective lens, and then the prism bends them so that they can come out through the oculars, come up the body tube into the ocular, be magnified, and then be visible. Little things like that, little improvements over time have made, it's still tedious work when you're using the microscope for hours at a time, but it's not as painful. Okay, so that's the body tube. That structure there is called the nose piece. It's a ring that rotates, and the objective lenses screw into this, so they're held pretty tight. The objective lenses here show up. We have the scanning lens. It's 4x. These are pretty consistent. The low power lens is 10x, and the high power lens is 40x. And then the oil immersion lens would say 100x. And I think it's been unscrewed off of this. Is usually on these nose pieces, there's a place for the fourth lens. So, okay, this is the mechanical stage. This is another big advantage. The mechanical stage works by moving up and down. That's how you focus. You focus the slide once it's in place by moving it up or down. So instead of having to, push it around yourself and try and find, move. Instead of old microscopes, you used to move the body tube up and down. And now you don't. You just the stage goes up and down. The stage also goes left and right and front and back, so you don't have to move the slide around by hand. Early anatomists had some real issues with. They had to do a lot of work just to look at something. So anyway, coarse adjustment, that moves the stage up and down fairly quickly. Fine adjustment is here, moves things up. We turn that around many, many times because you're causing minute changes to the stage. It deals with fine focus. The most microscopes now have a large outer dial and a smaller inner dial. The smaller inner dial is the fine adjustment, and the outer dial is the coarse adjustment. It's less mechanical wear and tear, I guess. I don't know. This hard-hitting structure is a light source. And adding the light to the microscope makes things a lot easier. Of course, the problem is that light bulbs burn out. And over time, you learn to be a pretty good expert in taking the microscopes apart and replacing light bulbs and fuses and stuff like that. So the light switch, here's the base, and the light switch is in the front on these. Every microscope is different. Some have the light switch on the side, some have the light switch in the back. The really, really nice ones that we use in microbiology have the light switch and the dimmer switch as one combined switch. You turn it on, and then you can increase the intensity of the light by turning the knob. So everybody's different. When you carry these things, you carry it by the base, one hand on the base, one hand on, this is called the arm. Two reasons. One, if you try to carry it by the arm only, they are heavy enough to drag your arm down a little bit. And the tendency is to swing your arm. And when you swing your arm, parts fly off. The ocular is not screwed in. It is just inserted in the tube and it can come flying out. And if the objective lenses aren't screwed on properly, they can come flying off. So it's just easier to carry it with your hand underneath the base and the other hand on the arm, if we were doing it on the ground. There is the iris diaphragm underneath. It regulates the amount of light. It's held in place by these little screws. And if you go to adjust the iris diaphragm and you're unscrewing it instead, the whole structure drops out in there, and then everybody panics, like, broke the microscope. No, it happens all the time. So sometimes they're pretty simple in how they're set up. Here's the diaphragm there. Now, close up on the diaphragm, this is the iris diaphragm. There's a lever here that allows you to open and shut the amount of light going through here. This is the light source. There's the fine adjustment. There's the coarse adjustment. This is the stage. This gives you a good view of the tip of the objective lenses. These clips hold the slide in place over the opening in the stage for the light to come through. I've seen people take microscope slides, and instead of putting them in here horizontally, I've seen them run them front and back. And then as soon as they move the stage, the slide breaks in pieces. I've seen just about anything, any mistake that can happen. It happens. So mistakes are going to happen. Okay. There's the mechanical stage. Back in the back here, these two control knobs that allow you to move the stage up and down and left and right. All the newer microscopes have them underneath. Makes it easier to reach, and they're not in the way of working here. Fine adjustment there. Diaphragm. Now this view from the top shows the stage and the control knobs. This little thing in here that looks like a lens is a lens. It's the condenser. It takes the light rays coming up from the light bulb underneath and focuses those. It actually condenses the light, and so that the light shines directly into the opening of the objective lens. It functions very much like a magnifying glass, that you can generate at some point. If you've ever played with a magnifying glass, you know that it can focus light into a very intense spot. And that's what this does here. This is called the condenser. These are the control knobs that allow you to allow the stage to go to the side to side, up at the front and back. Another view here. Here's your iris diaphragm. And your condenser is up inside here. And it gives you a nice look at the opening for the objective lens right there. So the lenses are set back, offset just a little bit, because people have been known to drive the objective lenses directly into a slide and break the slide. I mean, it happens all the time. You just you're cranking the stage and bringing it up, and you bring it up right into the objective lens. So we protect the important lens in the objective by having it set back just a little bit. Now, this is a good little chart to remember about magnifying power of the ocular. The ocular is always, the eyepiece is always has 10x magnification. The objective lenses are either going to be 4, 10, or 40 with a total magnification of 40, 100, or 400. So, resolving power. The microscope lets you see things that you can't see with your unassisted eyeball. The unassisted eyeball would, where the unassisted eyeball would see a straight line, we would see a line that is irregular and jagged. That is resolution, the ability to determine fine detail. Here's what I'm talking about. Take a look at this letter E. We said, ignore the fact it's upside down and backwards. I'll get to that in a second. Notice how irregular the edges are. This is what it looks like under 40 magnifications. 40 magnifications lets you see that from an inkjet, as the toner is applied to the paper, it leaves a distinct pattern, a splatter pattern, if you will, on the paper. You know, when we read something on a piece of paper with the letter E on it, it looks nice and sharp and crisp and clean, straight lines, and doesn't look like, you know, you drew it with a crayon like this does. But that's what it looks like in reality. But our eyeballs are not do not have the resolving power to see these various irregularities here. And so that's resolution. Resolution allows us to determine fine detail. And so the microscope allows us to see fine detail that we couldn't see normally. Now, the other thing that the microscope does is it inverts the picture and turns it upside down. Now, we don't care because cells are three-dimensional, you know, and we're not concerned that it's upside down or backwards. It all looks the same. You know, if you have a telescope, a good telescope, for example, and you observe the moon or a planet, your picture of your moon that you see through the telescope is going to be upside down and backwards. You take a photograph of it with your camera, then you can flip the picture in the computer to make it look the way it should. Because, you know, you're looking at it with the telescope, it's upside down, it's backwards, the craters are on the wrong side. You know, what you're seeing, so you flip it and put it the way it's supposed to. So with the microscope, what we're seeing in the microscope, it doesn't matter if it's upside down and backwards, because there really is no upside down and backwards for the cells. Okay, something else that we would see. If we were to take a specimen out of a human and put it on a microscope slide and try to look at it, we would probably not be able to see anything, because our tissues are, you know, underneath our skin. They all look the same. Everybody's tissues look exactly the same. They're mostly transparent. You know, even you know, we all have some organs are darker than others. Our livers and our hearts are dark. Our gallbladder are dark red. Our gallbladder is green in everybody. You know, our pancreas is sort of like a gray speckled structure. So it all looks the same. Our spleen is sort of purple. Everybody's organs look the same. Most of our tissues look light tan. Put them under the microscope and they all become transparent. So we use dyes, we use stains that alter the refraction of light that hits it. And so we can see structures much easier by staining the tissue. When you get into microbiology, you're going to discover that you need the stains to see the bacteria. Not only that, but stains can be used to identify what type of bacteria it is. You know, there's a staining process known as a gram stain, and if you're gram positive, you tend to using gram stain technique, the gram stain technique. If you are a positive bacteria, you stain purple. If you're gram negative, you stain pink, because it has to do with the type of carbohydrates and lipids found in the cell wall of the bacteria. And we categorize bacteria as either gram positive or gram negative. In fact, it's sort of a rule of thumb that a gram positive bacteria, if it stains purple, it's gram positive, and it's mostly going to be pathogenic. Gram negatives tend to not be pathogenic. They stain pink. It's not 100%, but it's a pretty good rule of thumb. Purple positive, pathogenic. So anyway, the staining allows you to look at tissues. Now, at this point in the lab, we would have taken a toothpick and scraped the inside of your cheek. I wouldn't. You do your own cheek. And then smear that on a microscope slide. That would be a specimen of human tissue. Now, we have dead cells being pushed up all the time on our bodies. In our mouths, we can scrape some of those off and look at them. But you wouldn't be able to see anything because it'd be transparent. But if you added a drop of a dye called methylene blue, everything would show up. And you could see the cell. You could see the nucleus of the cell. You could see the location of the cell membrane. You couldn't see the cell membrane, but you could see where it's located. We call it a wet mount. And this is for a lot of people, this is the first time to really use the microscope, and they're looking at your own cells. And it looks something like this. You would have a cluster of cells that stained blue with a dark blue nucleus. And the thing about the nucleus is that every nucleus is the same in your body. Every nucleus in every cell of your body has the same DNA. It's all the same DNA, every cell. Cheek cells have the DNA turned on just to be cheek cells, but everything else is there, brain and muscle and liver and kidney and all the other stuff. But it's just not turned on. And so you would see this. So the only cells in our bodies that don't have a nucleus are red blood cells, because red blood cells never divide. And this is what they actually, well, that was a professional one. This is what it often looks like, and for real. These are student slides here. So to prove it really does work. Here we have maybe 3 or 4 cells together, and there's a nucleus, and there's the cell membrane. There's the cytoplasm, the goop in the middle of the cell. You see it in 3 or 4 of them. Here's another one. Here's a cluster there that maybe not quite as focused as it could have been, but still had a cluster of cells. And every one of them stained purple, pink, stained blue. They're not pathogenic, not with methylene blue. They stain blue, and they all have a nucleus. So, they're not great slides, but it worked. This was a wet mount. Now, given our concerns over pathogens, even before the pandemic, these slides and the toothpicks and the cover slips and everything else associated with this would be thrown into the sharps box, where it would be incinerated later. So we were even before the pandemic, we would be very cautious about that. Okay, and that is everything on the microscope. Let me bring up one on those types of tissues that we were looking at. Here we go. Okay. The tissues I just showed you, the cheek cell slides, were called epithelial tissues. They are the most common type of tissues in the body. There are 4 distinct tissue types: epithelial cells, muscle cells, connective tissue, and nervous tissue. Epithelial covers all the exposed surfaces of the body. So all of our skin is all epithelial cells. Every opening into the body is epithelial. So the lining of your mouth, your entire digestive system from start to finish is lined with epithelial cells. The reproductive structures that are exposed to the outside are all lined with epithelial. Blood vessels are lined with endothelial cells. They are the same cells, but because they are internal, they're called endothelial. But epithelial cells cover the entire body, cover and line every opening. So they're probably the most abundant type of cell we have. Then we have 3 types of muscle cells, a half a dozen types of connective tissue, and 2 types of nerve cells. So let's take a look at these epithelial cells. 2 categories: simple and stratified. Simple is 1 cell layer thick. There are places where we want to have movement across the membrane of these cells, so we only have 1 cell layer thick. Stratified layers of epithelial cells occur where we have wear and tear, like inside your mouth. We routinely trash those cells. Those cells get worn away when we eat and drink. We want to continue to protect the lining of our mouth, so we have multiple layers, and they're just scraped off as we eat, scraped off as we drink, scraped off as we talk, and we push up new ones from underneath. Anywhere that we're going to have wear and tear and abrasion, we're going to have stratified layers. Our digestive system in many places has multiple layers. Our rectum and our anus lined with stratified epithelial cells. Where our skin is stratified, many layers of skin to protect us from wear and tear. That happens. So where we want to have movement of nutrients, for example, or movement of oxygen, we have 1 layer. Our kidneys have tubules made of simple layers of epithelial cells for movement of water when they filter out our blood. So we have simple and stratified. 3 types of cells: squamous epithelial cells. They are an irregular shaped cell. That's what you saw in the cheek cells. The cheek cells were squamous. They're flat. They have a big nucleus that sticks up in the middle. It really does protrude like a volcano in the middle, if you will. But the cell itself is fairly flat. Cuboidal cells are shaped like a cube, and columnar cells are shaped like a column. We also have 2 ringers. We have transitional epithelial. Those are located in the urinary bladder and the urethra and the ureters. They look like columnar cells, but when the bladder stretches out and the bladder fills up with urine, they end up looking like squamous cells. So they have the ability to stretch and change their shape. And then when the bladder empties and goes from the size of a grapefruit to the size of a walnut, then they go back to looking like columnar cells. Then the bladder fills up again and they stretch out and look like squamous cells. They go back and forth. So we call them transitional. And then we have the pseudostratified, meaning false stratification. They look like they're stratified, but they're not. And they have cilia. They have these little tiny hairs on there. And these hairs move in one direction. And we have them in various locations in our bodies. They play 2 key roles in movement. And we find them in our respiratory system, and we find them in the female reproductive system in the fallopian tube. Okay. This is simple squamous epithelial. Looks a lot like the cheek cells, except the cheek cells were stratified squamous. Stratified squamous, they're multi-layered because we have wear and tear. In the inside of our mouth, we eat, and we drink, and we talk, and we rip cells off all the time, so they have to be stratified. Simple squamous would show up in the lungs. The alveoli in the lungs where gas exchange takes place. One cell layer thick. They have imagine single ply wet toilet paper. That's the consistency of the alveoli. And they're made of simple squamous epithelial cells. Here are simple cuboidal cells. These form, these are what we're looking at are kidney tubules right here. Filtrate from our kidney goes through these tubules, and we want to recover good stuff, water and salts and glucose and other ions that are very important to us. So we want them to pass through these tubules while the waste products get trapped behind. And so we build our tubules out of simple cuboidal cells. We're looking straight down into a tubule, like looking down into a pipe or a smokestack, if you will, made of brick. Looking down into it, these cuboidal cells are stacked, in like a spiral shape inside. Better example here. Also notice a different type of stain was used on these cells. That pink and purple only applies, by the way, I mentioned to bacteria, not to regular cells. So these cells stained pink, and they're highly visible, but if we didn't have a stain, they would look transparent. Again, simple cuboidal, primarily in the kidneys to form kidney tubules. This is simple columnar. Simple columnar cells show up in our small intestine where we absorb nutrients. Small intestine is lined with simple columnar cells. One cell layer thick, right behind the simple columnar cells are capillaries, so that nutrients, after we've finished the digestion process of food in our stomach and in our duodenum, the first 12 inches of the small intestine, then we start absorbing the nutrients over the next 19 feet. And we line our stomach with simple columnar cells, and nutrients pass through and waste products don't. And they get absorbed here and they get passed into the capillaries, get into the blood supply. We also have to lubricate this area. We want to make sure there's no friction whatsoever. So we have these cells here called goblet cells. Goblet cells secrete mucus, and we have lots of goblet cells present so that material foodstuffs, digested foodstuffs, before it gets absorbed or as it's being absorbed, is lubricated. We've got lots of goblet cells in our mouth because that's where we call it mucous membrane. We have goblet cells lining our esophagus. We have goblet cells lining our small intestine, our large intestine, our rectum areas, because we don't like friction. Friction hurts. And so we have lots of mucus being generated by these goblet cells. And every one of these little structures here is a goblet cell. There, and there, and there, and there, and there. Okay. Here we see it again. Another picture of simple columnar cells. These are all one cell layer thick of simple columnar cells for nutrients that are found in here in the lumen, in the opening of the small intestine, to pass through these cells and go into the capillaries, which are back in here. And of course, these are all goblet cells secreting mucus to make the passage friction-free. Goblet cell. Now this is the pseudostratified ciliated columnar epithelial cells. They're columnar cells, but the way they stack on top of each other, they look like they're stratified, but they're not. They're simple, one layer thick, but they have cilia on top. They have lots of goblet cells lining this too. The cilia, these hairs, have the ability to move. They beat in one direction. Now, in this case, if this is in the trachea, which I suspect it is, these goblet cells here secrete lots of mucus. Mucus is used to line the trachea so that we can trap dust and dirt particles in here, contaminants that drift in as we breathe. Gets trapped and stuck in the mucus. The cilia move in one direction and they beat so that the mucus is dragged slowly up towards our throat and up to our oral cavity. And over the course of a 24-hour period, we drag up and swallow about a pint of that mucus, which goes into our stomach and is destroyed by stomach acid. And so we get rid of the contaminants that we inhale. The cilia are moving and working like that. Now, in the female reproductive tract, we see, same type of structure, but not as many, not as much mucus there. Same cilia. Notice all the simple columnar cells here. They look like they're stratified, but they're not. But here, what they're doing now is they're generating a means of propulsion for the egg. When a female ovulates her egg, the egg is released in the abdominal cavity and is drawn into the fallopian tube by the movement of the ends of the fallopian tube. They generate a current. Once in the tube, the egg has no ability to move on its own. And whether it gets fertilized or not, the egg is going to work its way down to the uterus. That's a 7-day trip, and it only has to go 2 inches. The cilia, which line the fallopian tube, beat in one direction. They go forward, and then they rebound. They go forward and rebound. And so they generate a very mild current that can propel that egg, whether it's fertilized or not, it will propel that egg to the uterus. Takes, like I said, takes 7 days to go 2 inches. So it's not exactly a blazing speed here. And that's what these cilia do. The cilia look like this. Notice how they're bent. Notice how they're bent with a curve there. What they do is they propel, they push in one direction and then they rebound where that curve is, and they reset themselves to do it again and again and again. And so these are going to push, in the case of the egg, they're going to push the egg, or drag the egg down to the uterus. It's the only way the egg moves. The egg has no ability to move on its own, and whether it's fertilized or whether it's not. This is the only way the egg gets down to the uterus. Okay. This is what's known as transitional epithelium. This is a urinary bladder. It looks like columnar cells when the bladder is small, but when the bladder expands, the bladder can go from something the size of a walnut to a grapefruit. It can end up holding about 1 liter of urine, stretched, fully stretched out. This expansive bladder gets bigger and bigger and bigger, and these columnar cells, which line the bladder, line the urethra, line the ureters, the tubes leading from the kidneys, will expand with it. And then when the urine is released, everything shrinks back down. It looks like columnar cells. They look like squamous cells when they're stretched and columnar when they're not. These are columnar cells here, but when they stretch out flat, they look like squamous. And this shows you, ureters, bladder, and part of the urethra. This is stratified squamous epithelial cells. You can't tell because they're in multi-layers, but this is what, if you had scraped the inside of your mouth, you'd be scraping these types of cells here. Stratified squamous cells, we find them in the mouth, we find them in the rectum, we find them in the anus, we find them in the vagina and the uterus, at the cervix, anywhere where we're going to have wear and tear in the body, we're going to have stratified squamous epithelial cells, multiple layers. And even though they're stratified, they still look just like a squamous cell, because the cheek cells look just like the simple squamous cells too. They all have that, squamous cells all have that irregular shape. Stratified squamous epithelium. The only difference between the external on the skin and internal on your mouth is whether or not they're going to be waterproof. You know, you're not going to have mucous membrane on your elbow. You're going to have the skin there is going to be filled with a waterproofing compound called keratin. You're not going to have keratin in your mouth or in your rectum or in the vagina or in the uterus. You're going to have mucous membrane there. So, stratified squamous epithelium. The non-keratinized versions show up in the esophagus, the mouth, the vagina, the urethra, the anus. The keratinized, the dry, waterproof version shows up on our skin. And that is epithelial tissues. Get into okay, we're almost done. We're on the home stretch now on this. So these are the remaining tissue types that we see: muscles, connective tissue, and nervous tissue. There's only 3 types of muscle cells. Only one is under voluntary control, and that's the striated or skeletal muscle. That's on the arms and legs, fingers and toes, hands and feet, stuff that allows us to manipulate our environment. They have stripes, light and dark bands. Under voluntary control, we can do things with skeletal muscle. Cardiac muscle is involuntary. The heart beats on its own, has no outside effects. The heart will beat on its own every 0.7 of a second. It is striated muscle. That's the only similarity it has to skeletal muscle. And of course, cardiac muscle is only found in the heart. But it, so it is striated, but it is involuntary. And then smooth muscle, totally different from the rest of them. It's not striated, it is involuntary, but it's shaped like a spindle. And it's only around our digestive system and our blood vessels and the hollow organs, like the outside of our bladder and stuff like that. So this is skeletal muscle. These are the striations, very distinctive imagery, light and dark bands here. And we'll talk all about that in chapter 9. Fat cells present here, lots of adipose tissue, lots of glycogen in skeletal muscle for energy. Skeletal muscle cells have multiple nuclei. We have lots of nuclei in here. It doesn't matter because the DNA never gets copied anyway, because muscle cells don't regenerate. Muscle cells don't undergo cell division or mitosis. So we don't need to worry about the DNA doing something unusual. All muscle cells can do is get bigger or smaller, weaker or stronger, depending on the amount of protein that and exercise they get. That's all. Under voluntary control, this is also responsible for most of our body heat. 60% of the energy that goes into muscle cells comes off as body heat. And that's what gives us our constant body temperature, thanks to our skeletal muscles. Smooth muscle is totally different. It's involuntary. We have no control over smooth muscle. The smooth muscle around our blood vessels, the smooth muscle around our digestive system that propel food through the digestive system. The smooth muscle around our blood vessels can cause the blood vessel to open up wide to increase blood flow, or to shrink down to reduce blood flow. Total involuntary actions on our part. We have no control over that. The central nervous system controls that instead. These are spindle-shaped. They also only have one nucleus. They too don't regenerate. There's a nucleus for each cell. They're pointing at both ends. So, again, smooth muscle. Let's see let me go back. Don't get this particular, come on, there we go. Don't get this picture confused with the picture of the stratified squamous. It looks very similar, but they're two separate things. You're going to see the giveaway is the shape of these nuclei. These flat nuclei are not present in that picture of the stratified squamous. Smooth muscle shows up in bands around the blood vessels and around the digestive system. I'll show you here. This is a cross section of the top of the small intestine, and right in here our digestive system, all 30-some feet of it, because in more or less, about 30 feet, lined with smooth muscle in two layers, a circular layer and a longitudinal layer that allows us to squeeze and propel the contents of the digestive system from start to finish, from the mouth to the anus. And so here we see the smooth muscle layers that surround our digestive tract. Now, cardiac muscles, totally different from anything else. Yes, they are striated, but they only have one nucleus, but they branch. They branch out from each other, and they have connections called intercalated discs. See, these cardiac muscle cells connect at the intercalated disc, and they have openings into each cell at the disc. And so what happens is when a electrical impulse goes into the heart, it shoots through these openings to every cell, because when the heart contracts, it's not like regular muscle. Skeletal muscle only stimulates the cells that are going to use for a specific action. You know, if you're driving a nail, you're going to use a lot of muscle cells in your arm. But if you're scratching your dog's ear, you're going to use less muscles. Depending on the activity, the type of action determines how many muscle cells that you're going to use. That's fine for skeletal muscle. The heart, though, when the heart contracts, every cell in the heart contracts together. There is no, well, not all of them got the word, or we're not going to contract everybody today. No, every cell in the heart contracts together, and they all contract with the exact same strength, the exact same force every time. So every 7/10 of a second, the heart's going to contract, and every muscle cell in the heart contracts together. And it does so because of these openings that connect between the cells. We branch and we connect at the intercalated disc. And so no other cell functions that way. None of our skeletal muscle cells, like the biceps or the triceps or anything like that, we control the strength of those muscles. But with the heart, it's everything. It's all or nothing. And another example here, you can see the striations, you can see the individual nuclei, and over here you can see very nicely the intercalated discs in through here. And this isn't so good because it's sort of like, well, there's one there and there's one there, but sometimes they're really hard to find, I'll just tell you that. But this is cardiac muscle right there. Striations, intercalated disc, and branches. Branching and discs really give away the cardiac muscle. That, in fact, they only have one nucleus per cell. And they don't regenerate either. They don't replicate either. The next category is connective tissue. Connective tissue are any tissues that meet the definition of living cells in a matrix that can be a solid, a liquid, or a gel with fibers embedded through the matrix. And that covers everything from bone to blood, everything in between. So you can have the matrix can be a solid, a liquid, or a gel with fibers there that may or may not be visible, with living cells stuck in matrix. The fibers that we encounter in here are going to be either collagen fibers or elastic fibers or reticular fibers. Collagen fibers are proteins. They are the most abundant protein in the body. They are the strongest fibers that we encounter. Each strand of collagen has the equivalent strength of a equivalent strand of stainless steel. So if you had a microscopic strand of stainless steel, a collagen fiber would be just as strong. Elastic fibers are made of elastin. How about that? They're stretchable. They allow our skin to rebound and resume its shape. And the reticular fibers form the framework, the mesh that helps hold our organs in place. They attach the epithelial cells to their basement membrane. They hold the viscera in place in the abdominal cavity. Collagen is very strong. The fibers in collagen can be lined up in parallel rows so that you have great strength with collagen. We use collagen in our bone to protect the bone from twisting and bending. Elastic fibers are stretchable. Our vocal cords are made of elastic fibers so that we can alter and change their shape and change the sound of our voice and change the intensity. The arteries are lined with elastic fibers. You'd notice that every time you take your pulse or take someone else's pulse. The pulse is nothing more than a surge of blood. You know, every time the heart beats, it generates a surge of blood out through the arteries. The artery wall expands as the surge of blood goes through there, and then it snaps back in place. That snapping back is the pulse. It's pulse and heartbeat are two separate things, but they count the same thing. They're counting the number of contractions the heart makes. So the pulse is just the snapping back of the artery wall. The trachea and our airway and the bronchi are all elastic. Our skin is very elastic. So we have this, elastic fibers that show up everywhere. And of course, the reticular fibers are the framework to hold things in place. Our covering of the spleen, because the spleen is such a squishy organ, it has the consistency of jello. And so to keep it from falling apart, we wrap it with this reticular fiber to give it its shape. The same applies to the liver. If you've ever seen raw liver, you know what raw liver is like. Well, that's what our liver is like. The reticular fibers give it its shape and keep it from just sort of oozing everywhere. This is what's known as loose connective tissue. Loose connective tissue has some collagen in it, some elastic in it, and some reticular fibers in it. The cells that are found in loose connective tissue are known as fibrocytes. They're made by fibroblasts. Fibrocytes and fibroblasts are what we see in scar tissue. They are the filler tissue. We use this as the packing material around our bodies to help hold organs in place. If you have a scar on your body anywhere, the scar comes from these fibroblasts that lay down fibrocytes. Scar tissue doesn't do anything except fill in the gap. It's a placeholder. It doesn't respond to touch, pressure, pain. It doesn't have hair on it. It doesn't contract. It's just there. There's a fibroblast nucleus. Elastic fiber, reticular fiber, well, they all look the same, don't they? You can't really tell looking at it what it is. All you can tell by looking at this is with all the fibers in there, it is loose connective tissue. It has living cells in a squishy matrix with lots of fibers. There's more nuclei there. Of course, there is a collagen fiber. Collagen fibers are, again, characteristic by having the fibers run row after row after row, side by side. Loose connective tissue has sort of an irregular shaped woven pattern here. And this is what it looks like here. Here is, again, this is loose connective tissue here. Here's our spleen. Wrap it around our spleen to help hold it in shape. It forms a internal soft skeleton, if you will. Now, dense connective tissue, which is much more organized, is what we see in our tendons and our ligaments, mostly collagen fibers. Lots and lots and lots of collagen fibers lined up side by side. Collagen fibers look like this. Collagen fibers have great strength because they have all these parallel rows of fibers, one after another after another. They form tendons that they start out, they come off of muscles as collagen fibers, and they form tendons. And the tendons attach to bone. Muscles don't attach to bones directly. They attach indirectly through tendons. So the muscles can pull on the tendons, and then the tendons pull on the bones. So we amplify the strength of the muscle, because muscles aren't strong enough to do their job without the bone. So we use the bone as a lever to get extra work done. And we attach with these tendons. Tendons are so strong that in most cases, they will snap the bone before they themselves break. The only exception to that is usually the Achilles tendon that attaches the gastrocnemius muscle, the calf muscle, to the calcaneus, or the heel of our foot. And so because that gets used all the time, we do see that snap occasionally. So it gets more abuse than any other tendon in the body. And the weave is very consistent, side by side by side. Imagine, if you had a thread and you wanted to pick up a 100-pound weight. Well, suppose you had, you went to pick it up with a single thread. Well, it's going to the thread's going to tear. But imagine 1,000 threads side by side attached to that weight at the same spot. You can easily pick up that 100-pound weight because they're all the loads being spread out, and they're all pulling together. And this is what we see in our tendons. Anywhere a muscle attaches to a bone, it's attaching through a tendon. Irregular connective tissue, like this, forms a capsule. So whereas dense regular connective has great strength in one direction, dense irregular has great strength in multiple directions because it forms a capsule around a joint. So we can have movement in various planes in here. Again, lots of collagen, a little elastic fibers in here too, some fibroblasts, mostly collagen. Great strength, but now we can move in different directions. Cartilage is a gel. It is a rubbery gel called chondrin. It gives us flexible support. Our nose is end of our nose is cartilage. Our trachea is cartilage. Our ears are cartilage. They have strength without a lot of weight, but they have flexibility too. They are laid down by what are cells known as chondroblasts. Anytime you have the suffix blast, it means builder. So chondroblast is a cartilage builder. we were originally, or as I probably said in the lecture earlier, our skeleton was in place 6 weeks after conception, not after birth, but after conception, all made of cartilage. And that became the framework for over the next 20 years for bone to form, instead ossification. And while when we're born, we have most of our bones in place, we still have a lot of cartilage. It actually takes about 23 years for us to fully ossify, calcify all of our bones. So anyway, so the cartilage looks like this. It looks like it's a gelatinous, it almost looks like plastic, like a squishy soft plastic with lots of bubbles in here. This is the chondrin laid down by the chondroblast. We find this at our knees, at the meniscus in our knee is made of cartilage. We find the ends of our bones are lined with cartilage to reduce friction when they're coming up against another bone. The chondroblasts lay down the matrix, the chondrin, but then they get trapped inside this bubble. At that point, they become chondrocytes. They are living cells. Whenever they have the suffix "cyte," it means a cell. So here we're seeing, this is hyaline cartilage, and these are chondrocytes inside their lacunae. That's what the bubble is called, lacunae. Now, cartilage has a problem in that they have no direct blood supply. No blood supply, no nerve supply. They get everything through diffusion, through the gel. So it takes longer for them to heal up when they're injured, as opposed to cells like bone, which have lots of blood supply. Because even though their cells are trapped too, I'll show you that here right next. So this is cartilage, living cells in a matrix with fibers, lots of elastic fibers in here, lots of collagen fibers. Here is bone. Bone has a hard matrix made of calcium and phosphorus and wrapped with collagen so that bone resists compression and twisting. With living cells, we have osteoblasts, again, builders, that are laying down the bony matrix. And as it hardens, bone cells get trapped inside these bubbles called lacunae. And those osteoblasts, those bone builders, blast is a builder, gets trapped and forms what are called osteocytes. The osteocytes are living bone cells, but they have the advantage over cartilage. All these little dark lines here are canals, and every osteocyte is in contact with every other osteocyte through extensions that run through these canals. And those all connect to blood vessels, arteries and veins, and nerves that are found in these central canals here. So these are living cells. So we have living cells in a matrix with fibers. Adipose tissue, connective tissue, living cells, there's a nucleus right there with elastic fibers wrapped around it. And blood. Blood meets the definition of connective tissue because you have living cells, you have red cells and white cells, and you have cell fragments called platelets. They're alive. The matrix is the plasma, the salt water that the red cells and white cells float in. Looks like this. This is the plasma in here. These are white cells. This is a white cell. This purple thing is a platelet. These are the red cells here. Platelet, white cell, and a bunch of red cells. Now, where are the fibers? The fibers are in solution. The fibers that are used to build a clot when there's an injury only show up when they become activated. So they only show up when there's trauma to a blood vessel, and then they form a clot. The rest of the time they are in solution, but blood cells meet the definition. Blood meets the definition of connective tissue because it has living cells in a matrix with fibers. These fibers that cause the clot are totally different than any other fiber that you encounter. Last tissue type, nervous cells. We see here, this is a great big neuron right in the center of the image here. And lots of these little purple things. These little purple things are called glial cells. These are all the support cells in here. A neuron communicates to other neurons by sending nerve impulses down this thing called the axon. It only goes in one direction. There's the cell body of the neuron. These purple things called neuroglial cells. There's about 8 different types of glial cells, and they do all the support work for the neuron. They provide nutrients, they fight off infections, they destroy worn out neurons, they initiate repair on some neurons not in the central nervous system. Because while neurons do not regenerate, we don't grow new nerve cells, they can repair themselves if the axon becomes damaged, but only in nerves that are outside the central nervous system. And it's a good thing to do because if we didn't have that, we couldn't do transplants. We couldn't do replacement. You know, when someone gets their finger cut off and has their finger put back on, we couldn't do that if we couldn't regenerate the axon at least. There's the nucleus of the neuron right there. Communication to the neuron only goes in one direction, it goes down the axon. So there's about 10 times more glial cells than there are neurons because they do all the support work for the neuron. So the neuron is free to do its sole job of communicating, sending signals back and forth. And they don't regenerate either. They do not undergo mitosis. We have all the neurons when we're born that we're ever going to have. We don't grow new neurons. Because we don't think we do. And we'll talk more about that when we get further along in the semester. So, this is an expanded view of that small intestine that I was talking about earlier. This shows an outer lid. Oh, wait, I didn't want to wait on this because we haven't talked about this in lecture yet. So we can address this next week. So I think I'll stop. There's only like 4 or 5 slides left, but I think I'll pick that up and cover that when I wrap up the lab this unit next week. But I want to cover some material in lecture. I haven't gotten that far yet in your lecture. So I'm going to stop right here. We only have a few slides to go, but I'd rather introduce the material in lecture than try it here. So, anybody have any questions on anything?
Yeah, what's the best way to, I guess, make this material stick? A lot of the stuff, at least to me, look very similar. Are there any kind of study guides to help differentiate?
There is a study guide in the table of contents in eLearn just for this first test, and it has pictures of all the important structures that you need to remember. I mean, yeah, a lot of it looks the same. And after sitting here for an hour and a half, and I keep throwing out pictures, it does look the same, doesn't it? You know, you can be honest, it's okay.
Do we have Quizlets?
Excuse me?
Do we have Quizlets?
No, you don't. I'm sorry.
That's fine.
So, and I don't have any practice exams either, but it'd be hard to do a practice exam. However, I would just go through the pictures, you know, and review them as often as possible. The study guide is going to give you a real good flavor of what to expect. If you know the parts of the microscope, there's a good portion of the test right there. But the study guide, I've put together a fairly detailed study guide just for this test. And it's in the table of contents in the eLearn. It's at the very bottom of the list of the content section. That should be a big help for you. And if you have other questions, I'll see if I can find other material. But if you use the study guide, you'll do well. I've seen that happen so many times. So.
And is that just for this first chapter? Or is that throughout?
It's just for this first unit test, because test 2 is on bones. You know, I'll give you a list of the bones you have to know, and there's all the pictures that go with on the PowerPoints. Yeah, I mean, I don't know how I could do a study guide for that, because it's just that's that type of thing. I've never done a study guide for the bone test. If I could find a good one, I would put it out for you. But I've looked for years and I've never found one that I liked. So, so on test 2 and 3 and 4, it's just lists of material. This one is a little bit different. Plus, this is the first test. I found historically that a lot of people, particularly on the ground, a lot of people never take the first test seriously and end up going badly on it. And I've been trying to break that habit for the past couple of years now, because I want people to do well. We're not in a class where I can say, hey, you need to stay and look at this. But you know, I really want people to do well in this first test, because people that do badly in the first test end up doing really well in the second test. But now you still got that, you know, other grade. The other thing I wanted to say is that as we move along, I intend to start putting out models and material in 217. If you're on camp, if you're in Sevierville, the Sevierville campus in Maples Marshall Hall, our lecture class and our lab class is designated as 217 up on the second floor. And I'm going to start putting out all the models, particularly when we get into the bones, out there for you to go in and take a look at, take out into the hall and study in there. Because I'm the only one that has it for classes and I'm not in there right now. So if you need to get in, if the door is locked, campus police will let you in, the office will let you in, and you can study in there and look at these bones. You really need to play with the bones to get the full flavor of it. You know, I can leave a couple of microscopes setting out in there if you want to go in and play with the microscope. But just, you know, anytime you're free, starting next week, that classroom will be open for you. So take advantage of that too if you get the chance. Any other questions? Okay, then I'm going to get us out of here. I hope I didn't put you to sleep too much on this. It will get better, trust me. It really will. So, and y'all have a good weekend. I'll see you next week.